Hello students, our next topic is neuroglia. As we have already seen in the previous class that the neurons cannot divide. Okay, whatever the number of neurons are present during the childhood, the same number will be continuing throughout the adulthood. Okay, throughout our life, that neurons are to be protected and they should be supported. These neurons are supported by means of the neuroglial cells. Here mainly you can see here the neurons of the CNS are supported and they are protected by means of the cells which are called as the neuroglial cells, and they will be more in number when compared with the neurons. But if we see them during our childhood, we can see that both are in same number. The neurons will be in the 86 billion will be there and the neuroglia will be 85 billion. But later on in the life, these nerve cells, we have seen that the neurons cannot divide. Okay, unlike the neurons, this neuroglia has the capacity to replicate throughout the life. That is, they'll be continuing to divide throughout the life and they'll be supporting or protecting the neurons. Okay, and here you can see that they are the non-excitable glial cells. Nothing but they cannot transmit or they cannot generate any impulse. Their main function is protection and supporting the neurons. That's all. They'll not do anything in the terms of impulse. Whenever any impulse is generated or whenever any impulse is transmitted, they do not perform any function. That's why they are called as the non-excitable glial cells, and they divide continuously throughout the life and they'll be protecting and supporting the neurons. And there are 4 types of the glial cells totally: astrocytes, oligodendrocytes, ependymal cells and the microglia. Let's see first about the astrocytes. Here, coming to the structure of the astrocytes, you can see here this is the astrocyte. Okay, these are the star shaped, roughly, not exact shape, they are the star shaped and they will be having, you can see the branches same as that of the dendrites. You can see here the branches, and this is the neuron. These are present around the neuron and they will be protecting the neuron. Okay, here see here, this is the mucopolysaccharide ground substance. Okay, see here, these astrocytes, they consist of a central nucleus and they are the star shaped and they'll be having the fine branches on them and they are lined on the mucopolysaccharide ground substance. This is nothing but, this consists of the mucopolysaccharides and proteins, and it is a gel-like substance. You can see here, it is a gel-like substance found mainly in the connective tissues, cartilage as well as the brain. Okay, that whatever the mucopolysaccharide substance present in the brain, there on that these astrocytes will be present. They'll be lying on this and they'll be supporting. They are the star shaped and they are having the fine branches, and their main thing is supportive tissue of the CNS. They will be supporting the neurons. And you can see here at the free ends, there are the bulges, the foot processes. Okay, these are called as the foot processes. You can see the small swellings are present on this. They are the foot processes of the astrocytes. And here astrocytes, along with this mucopolysaccharide substance, apart from supporting the neurons, they form a part of the blood-brain barrier. What is the blood-brain barrier? We all know that blood-brain barrier is a barrier or a protective membrane for the brain, which doesn't allow everything. Only the limited drugs or the limited substances will be entering through the blood-brain barrier, and it prevents the entry of the microbes or any antigens, and it prevents the infections also. This astrocytes plays an important role in the formation of the blood-brain barrier. Okay, this is about the astrocytes. They are the star shaped and at the end they are having the swellings which are called as the food processes, and they lie on the mucopolysaccharide ground substance, and their main thing is they are the supportive system for the neuron and they form a part of the blood-brain barrier. Okay, and coming to the next one, the second one, oligodendrocytes. Okay, where they are found means they are found in the nerve cell bodies. Okay, nerve cell bodies, mainly in the gray matter. We can see this. You can see here, mainly in the gray matter they are present. These are the oligodendrocytes. When compared with the astrocytes, they are smaller in size. Okay, and here the main function of this is the same, supportive function. And where they are present, here we are saying that it is forming the blood-brain barrier, or it is present near, lying in the mucopolysaccharide substance. These oligodendrocytes, they are present along the myelinated nerve fibers. See, and they help in protecting these Schwann cells also. Schwann cells are nothing but in the myelinated sheath only. The cells, we will be calling it as a Schwann cells. These oligodendrocytes will be supporting and they will be protecting these Schwann cells that are present on the myelinated nerve fibers, and these are smaller when compared with the astrocytes. The third one is the ependymal cells. As in the name itself, we can see that these are the epithelial lining. We all know that epithelial is nothing but a single layer of cells are present. We all know that epithelial lining is nothing but single layer of cells are present. These are the ependymal cells. These are the epithelial lining. Okay, they form the epithelial lining. Where means mainly the ventricles, and also in the central canal of spinal cord. Okay, they are formed as the cells form the epithelial lining of the ventricles of the brain, and also in the central canal of the spinal cord, and they also help in the secretion of the cerebrospinal fluid, which is present around the brain and the spinal cord. Okay, and here what are the main functions of these ependymal cells means they help in creating the brain cerebrospinal fluid barrier and helps in the secretion of the cerebrospinal fluid. And it also regulate how much amount of the cerebrospinal fluid is enough for the brain. It will also regulate the flow of the cerebrospinal fluid, and most of the wastage is present in the brain. How it should be removed? It also, these ependymal cells helps in clearing, helps in removing all the waste from the brain, and it also helps or plays a main role in the brain metabolism. Whatever drugs or any complex substance enters into the brain, it helps in the metabolism, that is break down into the simpler molecules. Whatever the molecules that enter into the brain, it helps in that. It helps mainly plays an important role in the brain metabolism. That is about the ependymal cells. They are the single epithelial lining that are formed in the ventricles of the brain and also in the central canal of the spinal cord. The next one is the microglia. We all know about the macrophages, right? Same as that of the macrophages, here the microglia will be performing the function. They are derived from the monocytes, mainly the cells that are present in the blood. Okay, from there they are formed, and before birth only they will be entering into the nervous system. They are not present in the blood vessels, but before birth only, from the blood mainly they are derived from the monocytes, and they will be entering into the nervous system. Okay, you can see here mainly in the nervous system, in the area of the blood vessels, you can see the microglial cells. Same as that of the macrophages only, they will be performing the function in the immune response. Immune response means whenever any antigen enters into a body, these microglial cells will enlarge. Okay, they'll increase in size and they become phagocytic. They remove that microbe and the damaged tissues, and they'll be engulfing that particular thing, and mainly in the area of the inflammation or in the cell damage areas, they'll be entering there and they'll be engulfing, they'll be removing all the microbes. Apart from that, apart from the immune response, they also show the brain development of the brain also. It performs a main function. It helps in repairing the injuries. Whenever small injuries are present in the brain, these microglial cells, they itself will help in repairing that injuries. And during the any inflammation, brain inflammation is there, they help in releasing the pro-inflammatory mediators or some of the markers in identifying the inflammation, and in knowing initial stages only it helps us in knowing about the neurological inflammation and helps us in diagnosing them. And apart from that, it also helps in supporting and protecting the CNS neurons. Okay, supports and protects the neurons. This is about all the neuroglial cells that are present. They are the main supporting system only. They do not help in transmission or impulse generation. Their main function is they help in supporting and protecting the neurons. These are the astrocytes, star shaped. Their main function is supporting. Oligodendrocytes, they are present in the myelinated nerve fibers. Their function is also supportive function. And ependymal cells mainly in the CSF and brain CSF barrier, and removes wastage from the brain and helps in the brain metabolism, regulates the flow of CSF. Microglia, we already seen this, helps in removing the microbes from our body, helps in removing the injuries, repairing the injuries, and also helps, it is an important mediator or marker in recognizing the neuroinflammation. And these neuroglial cells replicates, unlike the neuron cells, they continuously replicate throughout the life and protects the neurons. Thank you. Okay.
Sorry, she replied.